Thank you everyone for joining us for today's webinar. My name is Natalie Rodriguez and I am with the Forensic Technology Center of Excellence, a program of the National Institute of Justice. Before we get started, I will go over how you can view and interact with today's webinar. The first thing I'll draw your attention to is the chat pod, which is located on the right-hand side of your screen next to the player window. The chat is the best way to communicate any questions you have regarding today's material. All questions will be retained until the Q&A session after the presentation. The chat is also the best way to let our support team know if you are experiencing any technical issues. Simply post a short description of the issues you are having and our audience support team will assist you. If for some reason you happen to lose the audio or video during the live stream, the first thing you should do is refresh your web browser and then click the play button in the middle of the player window. If this does not work or you experience any other technical issues, please use the chat to notify our support team. You can also contact us with any customer support issues with the FTCOE toll-free number or email address located directly above the player window. We will begin today's webinar shortly, but first, please take a moment to type your location into the chat so we can get an idea of where everyone is joining us from today, as well as confirm that the chat is working properly. Welcome to the inaugural National Institute of Justice Forensic Science Graduate Research Symposium. Our first presenter is Teresa Tiedge, a PhD student at NC State University, presenting on the utilization of soil and dust eDNA to assist in criminal investigations. Without further delay, here's Teresa.
Well, thank you for the introduction. I'm just going to jump in here. So what is forensic geology? It is the application of geology to the investigation of a crime. And soil and dust are common geologic materials that are submitted to crime labs, and they contain geologic, biologic, and anthropogenic particles. And as with most forensic evidence, these materials are often submitted in trace amounts, such as a soil smear on a pair of jeans. And typically, forensic geologists will be asked to compare a question sample to a known. So does the soil from the crime scene match the soil that was found on the shovel in the suspect's car? And they will also be asked to determine provenance or sample origin. So did this soil come from New Jersey or did it come from Texas? And to answer these questions, forensic geologists typically use non-destructive or minimally destructive methods, which include color characterization with the Munsell color chart, measuring pH, using polarizing light microscopy to describe the optical properties of minerals, and X-ray diffraction to determine mineral crystal structure. And this is not an exhaustive list, and most of these analyses are not dictated by an SOP. Rather, they're performed on a sample-to-sample basis. And so the focus of geologic examinations is on the inorganic components, and the sample is typically consumed if it is small. And so there are a couple of limits when performing these analyses. The first being sample size. So bulk analysis methods require grams of soil, whereas less than 1 gram of soil may be submitted to the lab. And the second is that samples, they may be too similar for an inclusion or exclusion. And an example of this includes quartz-rich samples and soils with high organic content, both of which lack minerals. And also areas that have similar bedrock will have similar mineralogy, so reaching a conclusion might be challenging. Biologic components are present in these materials, but unfortunately often go unanalyzed, despite that associations between species and their environment is possible. And this is due to the lack of experts that are able to morphologically identify biological taxa, as well as the presence of partial fragments, which limits the expert's ability to identify the specimen. And so, based on these shortcomings, we asked, could DNA be used to analyze these materials more efficiently? And DNA metabarcoding is a widely used approach in the fields of ecology and microbiology, and it uses environmental DNA or eDNA from bulk sources such as soil, water, fecal material, and even dust. And so this approach involves amplifying and sequencing genomic barcodes, which are just short and informative regions of the genome that can be used to differentiate between species. And so this approach is also not limited by sample amount. Now in forensic science, DNA metabarcoding studies have mainly focused on the 16S barcode region of bacteria, and a few studies have looked at eukaryotes as a whole, or only plants or only fungi. No study has actually looked at bacteria, fungi, plants, and arthropods together. So DNA metabarcoding is therefore an attractive supplement for forensic geology investigations to capture additional evidentiary information and could be implemented most easily by labs already using next-generation sequencing for human casework. And so the overarching goal of this research is to determine whether bacteria, fungi, plants, and/or arthropods associated with soil and dust are stable for sample-to-sample comparisons and study site separation. And we define stability as the consistency and the persistence of a taxon. To achieve this goal, we had 4 aims. The first is to identify the most appropriate method to remove soil for eDNA analysis. The second is to collect mock geologic evidence from 2 study sites here in North Carolina. The third is to characterize biological taxa in soil and dust using DNA metabarcoding. And the fourth is to determine the stability of each taxon for sample-to-sample comparisons and study site separation. And so for today, I'll be discussing aims 1 through 3. For aim 1, we performed a soil isolation study where we picked 3 commonly used soil removal methods, which are swabbing, picking and scraping, and sonication. So we used mock evidence items, so the t-shirt, boot sole, and trowels to complete this study. And soil was rubbed into each of these items and remained in contact with the soil for about 2 weeks. And one of each mock evidence type was subjected to each removal method and sampled in triplicate. The DNA was isolated using the Power Soil Pro Kit and then quantified via the qubit. And so here we have the average DNA concentrations and standard deviations for each soil removal method and mock evidence item. And so we can see here that picking and scraping yielded the most amount of DNA and was statistically significant compared to the other methods when we applied a t-test. And so considering these large differences in concentration, we were curious to see if either method would impact which taxa we recovered. And so to assess that, we used DNA metabarcoding, where we performed amplification using these barcode regions that have been published in the literature. And after amplification, we manually prepared libraries using a library prep kit, quantified our libraries with qPCR, and then sequenced them on the mini-seq. And so after bioinformatic processing, we looked at the number of unique amplicon sequence variants for each mock evidence type, and displayed here are the results for the trowels. So we looked at 2 replicates for each removal method and for each barcoding region. And we found that within each category, the number of ASVs here in the blue remained consistent. So although not displayed, these results were very similar for t-shirts as well as for boot soles. And we performed an ANOVA and applied the Tukey-Kramer on a significance test for multiple comparisons, and the total number of ASVs was not significant between methods or mock evidence item. So based on these combined results, we decided on scraping as our method for aim 2. And so here, we wanted to assess how spatial and temporal variables, as well as mock evidence type, could affect taxon stability. So with this in mind, we selected 2 locations here in Raleigh, North Carolina, an agricultural location and an urban location, where we could collect mock evidence monthly for a 1-year period to capture changes in both temperature and precipitation. And so for soil, our mock evidence included surface soil that was rubbed onto t-shirts, boot soles, and trowels, and we also collected reference soil cores from each location. For dust, our mock evidence included exposed dust on brick pavers, as well as dry dust from air filters. So in March 2021, all of our mock evidence was placed into these dog crates at each location to minimize sample loss. And so each item was collected monthly. And so here you can see what the cage looks like for the ag site as well as the urban site. We had 2 crates set up at each site, so we collected 2 of each mock evidence item per location every month. And so here on the right, we have a schematic of our site setup. In the center here, we have our 1 cage. Then approximately 10 meters to the right, we have our second cage. We also collected soil cores approximately 10 meters in the remaining cardinal directions to assess site variability. And we started these collections in May of 2021 and completed all collections in April of 2022. So moving into the experimental design for AIM-3, which is the characterization of biological taxa, we took our soil and dust samples and extracted the DNA using the Power Soil Pro Kit, and total DNA was then quantified via the qubit. So next, for amplification, we use custom indexed PCR primers that contain the Illumina adapter sequences and amplified 16S for bacteria, ITS1 for fungi, ITS2 and tRNL for plants, and CO1 for arthropods, and this was all performed in duplicate. So unlike the metabarcoding workflow I described for the soil isolation study, where we performed amplification and library prep separately, PCR and library prep are done concurrently at this step. So following amplification, libraries are then quantified via qPCR, and then we send our libraries to the core facility here at NC State for sequencing on the MiSeq. When we get that data back, we demultiplex our reads using QIIME2, and then trim reads using CutAdapt. Then we identify our unique amplicon sequence variants, or ASVs, via data2, and then these unique ASVs are then searched against GenBank for taxonomic identification. Once we have all of our data, we'll then perform the statistical analyses at the end. So here, I just wanted to show you some examples of mock evidence that I've collected over the year. We have the trowel, soil removed from this boot sole. We have a t-shirt, the soil cores collected from one location, and then a cutting of the air filter. I also wanted to mention that we determined the pH of each of our soil cores as well. For sample processing, the DNA has been isolated and quantified for all 1,026 samples in this project. And here on the right, I have a plot of all of our DNA concentrations for soil displayed in order of collection time. We observed that DNA yield was the highest in the agricultural samples and was more variable compared to the urban samples down here at the bottom. So these next few slides are some preliminary assessments and use a small number of samples. So to orient you to this figure, the graph is organized with the agricultural samples in the top panel and the urban samples down here at the bottom. These samples are from May, and they are organized by mock evidence type. This data here is looking at tRNL abundance, and the first thing I wanted to highlight is that the composition is different between these two locations. Additionally, the boot sole has a different plant composition compared to the shirt and the trowel, and the shirt and the trowel are pretty consistent with each other, but they do have differences in abundance. Now, if we move over to the right side here, again, our samples are separated by location. But here we're looking at soil cores from May and from June. And so the plant composition does differ, again, between the two sites, but the composition is pretty consistent between the 2 months, but we do see a difference in abundance. The next figure is 16S abundance. And so here we see that both sites are pretty similar to each other and that the bacterial composition is consistent between mock evidence items. And when we're looking at the cores, again, we see that sites are similar, and from May to June, the composition seems relatively unchanged. However, both of these panels do show differences in abundance. And lastly, here we're looking at ITS1 abundance, and we see the same results for fungi as we did with bacteria, where the fungal composition is consistent across mock evidence item and is relatively consistent between sites. And again, we do see those shifts in abundance. And so I wanted to end this presentation with some conclusions, which are picking and scraping is an appropriate method for eDNA analysis. 100 milligram soil samples yielded enough DNA for analysis. And sequencing eDNA from geologic materials using custom index primers is possible. And as far as finishing this research project, I would like to complete the remaining amplifications and DNA sequencing, and then determine the stability for each taxon. And so with that, I would like to thank you for your time. And if you have any questions, please put them in the chat.
Thank you, Teresa. Our next presenter is Petra Banks, a doctoral candidate at Texas State University, presenting a comparison of skeletal trauma between blast trauma and trauma from falls. Take it away, Petra.
Hello, my name is Petra Banks, and I'm presenting skeletal blast trauma, determining the effect of known and experimental blast events on trauma patterns and fracture behavior. So this is as part of my dissertation that's funded through NIJ. So the purpose of this dissertation is to identify blast trauma patterns, looking at blast events, accidental, suicidal, homicidal, or warfare, and potentially similar trauma that may mimic blast events like falls, building collapse, vehicular impacts, and aircraft crashes. I'm looking at this data through medical examiners and coroners' offices and the Defense POW MIA Accounting Agency, DPAA. I'm also looking at experimental blast trauma to try and narrow down that blast wave impact to look at one aspect through the Forensic Anthropology Center at Texas State, FACTS, using their Willed Body Donation Program. So a little background on explosions. There's different types of explosions. There's high explosives and low explosives. High explosives create detonation, so this creates brisance and is characterized by a supersonic blast wave that moves outward from the point of origin. Low explosives generate deflagration, so this is fire. They don't typically have brisance, and they're subsonic with no blast wave. So the difference basically is that blast wave itself. You can have low explosives that will generate into a detonation, but that's only if they're really highly contained. So things that are high explosives are things like C4, so this will automatically detonate. Things like fireworks or movie explosions are deflagration, but they can generate into a high explosive if they're contained well enough. The images to the right, you'll see Los Alamos National Laboratory's test explosion, and then on the bottom is from the movie Spectre. This was the Guinness World Record of explosions in movies. And you can see the difference in the fire in the bottom versus kind of that flash in the top. Okay, so when we look at blast trauma, there's different types of blast trauma. The first type is primary. This is caused by the blast wave itself. And if you look at the image on the left, there's a period of positive pressure that's generated as the air compresses as it leaves the origin. And that high pressure is immediately followed by a section of low pressure that extends for a longer period of time. And typically, any trauma that happens due to this pressure change is related to air pockets in the bodies. You'll have things like eardrums or lungs or bowels that will have those air pockets expand, causing trauma. There is skeletal trauma associated with this as well, but it's not particularly well studied. Secondary blast trauma is caused by fragmentation or shrapnel, either fragmentation from the casing or shrapnel or any object between the person and the origin of the blast. Tertiary blast trauma is caused by the body impacting other objects. So if a body is thrown in the air and hits a surface or is knocked down, that is tertiary trauma. And quaternary is indirect injuries caused by burn, smoke inhalation, or building collapse, etc. And these, I'm looking at all of these when I'm looking at records from offices because there's no way to just distinguish from one unless I've got really detailed data on the blast event, but typically that is not available. So I've got all three or all four. Okay, so my research questions are above. So research question 1 is what differences exist in skeletal blast trauma location, type, and frequency between accidental and intentional blast events and other traumas? And how do these civilian traumas compare to skeletal blast trauma and other trauma from warfare and experimental blasts? So this will be what's discussed today. Research question 2 and 3 are not going to be covered, but I'll read them out anyway. Research question 2 is when intrinsic and extrinsic factors are accounted for, what is the typical fracture behavior, type, and location for blast trauma caused by the blast wave? And 3 is, are current methods of determining direction and type of skeletal trauma caused by a blast wave accurate? So those last 2 will be answered through the experimental research, which hasn't been conducted yet. Okay, so looking at research question 1. So the methods for this are collecting data from medical examiners and coroner's offices, typically autopsy reports, and then some data about the event itself, and then data from DPAA. For the purposes of the research today that I'm going to be presenting, I'm only looking at one office at the moment, and only looking at blast events and falls and comparing those two. So on the right, you can see this is my access database form. You've got the individual record information right here in the center, and then trauma data to the right. Individual record will include data about the event itself, as well as some basic demographics, so I can examine that further as I get into the data processing. Okay, and then when you click those 2 boxes on the right in this last image, you get trauma location and trauma type. So trauma location breaks out into general body part location, and then more specifically, also looking at each skeletal element, and then zones within those affected elements. So the zones are using a CORA, which is a system developed by DPAA. They've broken each bone into zones, so I'm using those. And then I'm also looking at trauma type. And you can see up here on the top left that it says trauma ID. So an individual might have multiple trauma IDs, and each trauma ID might have multiple types and multiple areas affected. Typically, it's all going to be in one area. So you'll have like a shoulder, and that might have soft tissue affected as well as skeletal elements affected. So I'd be recording both of those in this trauma location information. And then also things like laceration and, fracture, anything else. Oftentimes, there's not a whole lot of detail in the fracture. So this second column is typically just left undetermined or general fracture. Okay. So the preliminary results of the fall and the blast, when I first ran it, I obviously was looking at counts. That was the easiest way to do it, and kind of broke it into general body areas. You can see on the right, is blasts is 68 individuals and falls are 15. So to try and adjust for that, I created a percentage system, just looking at percentage of blast trauma that's occurring, rather than trying to look at it from like a count so that the falls aren't being diminished by that. And when we look at this, you see that there's consistently the head and the ribs are the most affected in both cases. However, when you look at falls, they tend to be more uniform across the rest of the body, particularly in the rest of the torso. So the spine, the pelvis, and the neck are all affected, and the arms are more affected than the legs. Strangely, in blasts, the legs are more affected than the arms. So I have some ideas about why that might be. This is obviously very preliminary, so I don't know exactly what's going on yet, and there's going to be a lot more data processing that will take place that incorporates all those details that I'm collecting. But at this point, we're just looking at the kind of basic trends. So when we look at the falls are affecting more parts of the body, and this is likely because when a body is falling, that they're going to impact the ground at a consistent force, right? So the whole body is impacting the ground. When you look at blast trauma, a lot of the cases have smaller explosives involved, and so they affect the body in tight areas rather than a full body impact. Some cases are not that way, so you still have more unified effects in some cases, but for the most part, it's going to be a little more localized, especially with civilian trauma. And the other factor that's important to consider is that even if you have an explosive that's a little bit larger, if it's far away from a body, it's going to have a high dissipation factor. So the wave itself dissipates rapidly. So if it's against the body, you have absolute destruction, but if it's 2 feet away, even if it's like C4 or something like that, that's high detonation, it will dissipate so fast that you will have fractures and maybe death, but you're not going to have nearly the same effect that you have if it was right up against the body. So the limitations that I'm dealing with, a blast event causes are variable. So we're looking at anything from a gas explosion to a firework that I've got in my data collection, or a terrorist attack in some cases, like a car bomb kind of setup. So these can be very different events. So I've got some of the details that are recorded for that I can calibrate for in the future, but it's something that's going to have to be considered in the future research. The blast events can also be complicated by building collapse and fires. So when a building collapses, you're going to have very different results than if a building doesn't collapse. Same with fires. And falls, when we think about falls, they can result in variable trauma depending on the position of the body on the impact. So we have lots of arm impact and chest and torso, which kind of implies a certain way of falling. But it's hard to say like what would happen if, you know, if different cases, like this might be more true for falls in a city or one type of event versus like falls for people that are, you know, skydivers or something and have a failed shoot. They might be landing differently. All right, so thanks to the National Institute of Justice for their funding, my advisor, Dr. Nick Herman, my committee, Drs. Kate Spradley, Danny Westcott, and Jason Worsema, the Freeman Fellows and Phi Kappa Phi for their funding, and the Facts Will Body Donation Program donors and their families for their generous contribution to this research. And here's my reference screen. Please feel free to scan the QR code. Don't be thrown off by it being my proposal as well, but it's very detailed and should be helpful to you if you're interested in more information about this research. And if you have any questions, please feel free to put them into the chat. Thank you.
Thank you, Petra. Our final presenter of Session 1 is Dr. Joseph Cox, a postdoctoral associate at Florida International University, presenting on quantitation of fentanyl and metabolites from blowfly tissue and development effects of fentanyl on Lucilia sericata. Here is Joseph.
Thank you for that introduction. Today I was talking about the quantitation of fentanyl and metabolites from blowfly tissue. Before we begin, I want to have a quick disclaimer. Digging into the presentation, we first need to talk about forensic entomology and what that is. So the study of insects and other arthropods during a criminal investigation, predominantly used in investigations involving human corpses. This is done by calculating the minimum postmortem interval, or min PMI. Beyond 72 hours, forensic entomology is one of the most reliable ways to calculate the postmortem interval. And this is done by calculating the minimum period of time required for insects to develop. We're able to do this because insects grow at a very temperature-dependent rate. So if we know the weather conditions at the time of death, and we collect insects from the scene, we can calculate the minimum time that is required, the insects are required to reach that stage, and therefore the minimum time that a body could have been exposed to the elements. Insects grow at a very predictable life cycle. They begin as deposited eggs on the body. They quickly emerge and grow to first instars, molt, become second instars, continue feeding, become third instar, and then begin to wander. They wander away from the corpse, they stop feeding. This behavioral change is due to them begin to want to pupate. When they pupate, they have a hard exoskeleton which protects them from the elements. However, it doesn't protect them for predation, so they need to find a safe place to pupate. Once pupation is complete, they emerge as adults and leave behind an ictuviparium. So digging into the core of this presentation is focusing in on forensic entomotoxicology, which is a specific field of entomology that focuses in on insects as a toxicology matrix, as well as the effects of drugs or toxins on insects themselves. This relies on the accumulation of drugs or pollutants in the insect tissue, and is focused in on the developmental effects, as well as insects as a toxicology matrix. Drugs have been shown to have an impact on insect growth and development, and that's important because that can affect the PMI. So antemortem ingestion of drugs by the decedent has been shown to slow down or speed up development in insects. Cocaine and methamphetamine have been studied to show increased growth rates in insects compared with insects feeding on cocaine-infused tissues showing a faster rate of development compared to control groups, as well as methamphetamine showing an increase to third instar as well as to pupation. Methylphenidate, morphine, and heroin have been the opposite effects. They slow down the growth and development of the insects. With methylphenidate slowing it down by 24 hours and morphine and heroin by 29 hours to third instar larvae. So some typical toxicology matrices include blood, either central peripheral, liver tissue, or other fluids in terms of advanced decomposition. But insects can also be used as matrices. So when the drugs are incorporated into the insect tissue as the insects feed, and this can be a valuable tool, especially in terms of cases with advanced decomposition. insect specimens persist in the environment over long periods of time and provide an additional matrix, especially the harder shells, so the pupa and the enteroperium, those persist in the environment for much longer than soft tissue or blood. So some hypotheses for the study: if fentanyl is present in the feeding media, then this will impact the growth and development of insects. If fentanyl is present in the feeding media, then fentanyl or metabolites will be detected in all the life stages of the insects. And if fentanyl is detected in the insect tissue, then a correlation of the feeding tissue, which is liver in this instance, to the fentanyl concentration in the insect will be determined. We also had a unique opportunity in this study, is that we had an authentic postmortem liver sample with fentanyl, norfentanyl, and 4-AMP concentrations in the actual tissue. This allowed us to compare our treatments to this tissue as a validation set for our growth curves, our toxicology estimations, as well as insect stage comparisons. So the overall project roadmap, we began with our extraction, our method development. We validated that method by the ASB 036. And then for our overall experiment, we compared physical characteristics of each treatment as well as our toxicology results. We began with our extraction process with homogenization. And it is very important to homogenize and crush the insect tissue to allow for extraction of the drugs. We performed this using steel balls, our homemade reaction chamber, as well as an MM200 for high-speed impact pulverization. This allowed for the insects to be homogenized as well as ground up so that way we can extract them. Our extraction process was a catcher's extraction, which is quick, easy, cheap, effective, rugged, and safe. We used the Agilent bond elute extraction kit. We modified that down to our small extraction size with approximately 0.2 grams of insect tissue, petrosalt, acetonitrile, and water. We mixed all that together with the mixer mill, centrifuged that down, removed our supernatant, cleaned up the supernatant using dispersive SP, and then dried down and reconstituted that for LCMS analysis. We validated using the ASB 036, which includes all of these aspects, but we're only going to focus for time, we're going to focus on the matrix effects and bias precision for the larvae, but this is performed for all the insect tissue. Matrix effects is performed by comparing ionization, suppression, and enhancement at a low and a high concentration for 10 replicates. Shown here is the larvae. Plus or minus 25% is deemed acceptable by the ASB, and did not exceed 17.9% at both concentrations for all four of the analytes we observed. So fentanyl, as well as the metabolized 4-MVP, norfentanyl, beta-hydroxyfentanyl. We also evaluated bias and precision. This was done in triplicate over 5 runs for a total of 15 replicates. As shown here, 20% CV is deemed acceptable, and for the within run precision, we did not exceed 10.8. For the between run precision, we did not exceed 12.2%. And for bias and precision, the ASB deems plus or minus 20% acceptable, and we did not exceed 10.1%. It's performed for all tissue of our analysis, including liver, larvae, pupa, enteroperium, and adults. The overall design, we placed 70 to 85 eggs per treatment over 4 treatments of a 0, which is our control group, 10, 100, and 350 micrograms per kilogram of fentanyl into the liver. This is our 4 treatments over 7 days of collection for 3 replicates and for 70 to 85 eggs. Shown here on the right is our Percival I-36 incubation chamber, and below we controlled for temperature, the light-dark ratio, as well as humidity. The concentrations of fentanyl, which shows based off of literature review, with the 10 being a non-fentanyl, a non-drug-related fentanyl overdose, 100 being an average, and 350 being the high. So to create our replicates, we homogenized 200 grams of human liver and spiked those to the concentrations we deemed. So we had control groups 10, 100, and 350. So once we homogenized them, we aliquoted out 25 grams of specimen, and then we placed 70 to 85 eggs on each one. So each one of the 7 replicates represented a day of collection: 2, 3, 4, 5, 6, 10, and 21. The entire cohort was collected, and then we performed data analysis. This is performed for each one of the treatment groups. Within that, so each one of that represented a replicate. This was replicated 3 times over a period of 3 months. And then we collected all the insects and we extracted them. So we randomly selected single larvae as well as we randomly selected pooled larvae. So we pooled larvae, we analyzed the toxicology specimen as a single larvae shown here on the left and a pooled larvae shown on the right. Over the treatments, we observed fentanyl in all the treatments, low, medium, and high, and they increased as the concentration of the liver increased, the concentration in the insect also increased. Norfentanyl was only observed in the medium and high treatments, as well as 4-MPP and beta-hydroxyfentanyl were not observed in the day 3 feeding larvae. Correlation was determined from the larvae concentration to the liver concentration, shown here. On the left is a single, and on the right is the pooled, with the liver concentration on the X and the larvae concentration of fentanyl on the Y. We had R-squared values of greater than 0.9 for all of the replicates. Shown here are the pooled for a single, and they were 0.928 for the single and 0.988 for the pooled specimens, showing a high correlation between the larvae and liver concentrations. For the validation set, or the authentic liver set, shown here, the fentanyl concentrations for the larvae that we extracted were similar to the medium treatment. The medium treatment was 100 micrograms per kilogram of fentanyl, and the authentic set was 111 micrograms per kilogram. So having a similar concentration was expected. The concentration for the authentic set of norfentanyl was higher than that of the medium treatment. This is because the norfentanyl concentration had a higher concentration already, because the treatments did not have anything besides fentanyl in them. There was no metabolites placed, so any metabolism or any metabolites detected is an indication of metabolism by the insect. The same with 4-MPP. There was no 4-MPP observed in any of the larvae. However, there was 4-MPP observed in the authentic set. That was because 4-MPP was present in the liver tissue. Using the authentic set, the concentration of the larvae were used with the linear regression from the treatments to calculate an estimation of liver concentration based on the larvae concentration of the authentic set. This produced a average concentration, estimated liver concentration of 87. If you remember, the concentration was 111 micrograms per kilogram. So it was a bias of negative 21% for the single larvae. For the pooled larvae, it was a bias of negative 32%. However, the single larvae did have one sample that was much lower in concentration, and if that was removed, then it was negative 11%. For the day 6 larvae, there was much lower concentrations of fentanyl detected in the larvae, only in the high for the single, only in the medium, and norfentanyl was observed in the medium and high concentrations. Beta-hydroxyfentanyl, however, was observed in the medium and high treatments for the day 6 larvae, which was different from the day 3 feeding larvae. In the pupa, fentanyl was observed in very low concentration only in the high for the pooled. However, norfentanyl was observed in the low, medium, and high concentrations, as well as beta-hydroxyfentanyl observed in the high and in the medium for the pooled. For the later stage of the infant-aquarium and the adult flies, fentanyl was observed in the low, medium, and high treatments. Norfentanyl was observed in the medium and high treatments, and beta-hydroxyfentanyl was observed in all 3 of the treatments. In the adult flies, fentanyl was observed in all 3 of the treatments, as well as norfentanyl. However, beta-hydroxyfentanyl was not observed. This could be indicative that beta-hydroxyfentanyl is left behind in the aquarium and is not present in the adults. So for the insect metabolism, in human metabolism, the predominant metabolites are norfentanyl, 4-MPP, and with the minor beta-hydroxy. However, in the insects, 4-MPP was not observed. So for the physical characteristics, I'm going to briefly go through this, but shown here, we collected larvae, pupa, and adult flies. We measured them using a microscope. We observed percent stages. So for day 4, highlighting day 4, we have going down, we have low, the control, low, medium, and high, and the authentic set. At day 4, the authentic liver, or the authentic specimens, insects were 88% third instar larvae compared to the control group only being 28% third instar feeding larvae. At day 5, all the insects were wandering, all the insect treatment groups were wandering. The authentic set was behind with only 41% of the insects still wandering. In the pupa, by day 10, in the control group, 80% of the insects were pupa, and the treatment groups were fairly similar, with the authentic set being 68%. And the adults were all fairly similar percentage of being adults by day 21. For the length presented here, the control group shown here in blue, the high group, treatment group shown here in red, low, medium, and then our authentic set. As you can see here, our error bars, we had a very high amount of differences in our groups. So there was a lot of variation. However, the trend of the control group matches a very typical growth curve for the insects. However, the treatment groups had a very odd shape for the control, for the growth curves, indicating that there was some impact of the drug on the growth of the insects. However, it was not a pattern that could be discerned. So there was differences in the various control groups or various treatment groups compared to the control groups for the growth. However, it lacked a consistent trend, so no significant statistical inferences could be drawn. We were able to quantitate fentanyl metabolites from all the life stages. Specifically, the late stage of improperium has great value because these are hard, exoskeleton stays behind. We observed metabolism of fentanyl to norfentanyl and beta-hydroxyfentanyl. However, no form V was observed. There was a high correlation for the liver tissue to the feeding larvae tissues, and we were able to estimate liver concentrations based on the insect concentration in the validation set. And the fentanyl did impact the growth of the insects. However, the pattern was not discernible. There's a lot of strength in this design as it gives insight across all the life stages. It established persistence of the drugs throughout all the life stages and allows for future work to focus in on specific life stages, as well as application to different insect species, drugs, and other combinations. I'd like to acknowledge my research group, my committee, of course, the Department of Forensic Science at WU, Dr. Craner, Dr. Newman, WB Gift Registry, as well as the National Institute of Justice for funding. And I look forward to any questions that you have. Also shown here is the link for my dissertation, so you can dig into there if you have any further questions after this presentation. Thank you.
Hello, my name is Teresa Johnson, and I'm a physical scientist with the National Institute of Justice Office of Investigative and Forensic Science. I'm going to say thank you very much to Teresa, Petra, and Joseph for providing such informative presentations. And we will now begin with our first Q&A session. So if you have questions for our speakers, please put them in the chat and make a note for who it is. We are going to start with a question for Teresa. So Teresa, in addition to eDNA from bacteria, arthropods, plants, or fungi, what other kinds of information do you think can be gleaned from extracted soil and dust samples for forensic purposes?
That's a great question. Specifically for biological components, I think potentially looking at RNA would be very interesting, so the transcriptome of the soil and dust. Also proteins as well, so the proteome. I think that could be an interesting approach to take. And also we didn't focus on viruses, but potentially looking at viruses themselves and seeing just the viral composition found within soil and dust would be a unique approach as well.
Okay, thank you. Just a note to folks, Teresa and Petra and Joseph, if you could turn their cameras on, that would be fantastic. Thank you. All right, so the next question I have is for Petra. Petra, how does the skeletal trauma documentation and the analysis or interpretation process differ for decedents in different stages of decomposition, such as, you know, recent death versus something where you have only skeletal remains?
Yeah, so part of the way I'm accounting for that is looking at missing elements. So one of the biggest differences between a skeletal remain recovery and fresh body recovery is going to be the missing elements that occur. You know, hands and feet are often lost in that process of decomposition. And so I'm recording missing elements as well. That's also the case for like even a fresh body in like an aircraft crash is going to have potentially missing trauma. However, the rest of the trauma analysis should be fairly clear regardless of decomposition. There's not a whole lot of alteration that occurs in the fracture surfaces and the fracture shapes themselves. So as long as I have detailed data, it's going to be actually better in the skeletal analysis because that means a forensic anthropologist has looked at it versus fresh trauma where typically an ME or coroner is only going to record that there was a fracture in an area. They may not be recording exact types of fracture, which is more common for a forensic anthropologist to assess. So they'll talk about like a butterfly fracture or a comminuted fracture in a little more detail than you'll get from an ME. So yeah, it's pretty equal for the most part.
Thanks for that, Petra. Joseph, how do you think the larval growth cycle would be affected by polydrug use or the presence of drug metabolites?
That's a very good question. It could have a big impact. As I showed in the beginning of my presentation, that some analytes speed up growth and some analytes slow down growth. So there could be either synergistic if they match the same drug, or it could be kind of a balancing if they're counteractive. So yeah, it could have a big impact. That's one of the reasons why I was trying to focus in on just one drug by spiking one drug into the livers. But actually, my authentic set, my validation set, was way behind when it came to growth. And actually, I only evaluated specifically for fentanyl metabolites to test for that. I don't know actually if there were other drugs present, so that could actually be a factor in why the growth rate was different for that validation set. Because I didn't do like a non-target analysis. I don't know specifically what else could it be in that liver.
Understood. Yes. All right. So circling back to Teresa, I have a quick question. Can you talk us through maybe what the differences were in eDNA across the sample types? I found that was fairly interesting. That the T-shirt, was different than say the shovel or the sole of the shoe. I understand obviously there are going to be some seasonal, some locational differences. I guess I didn't anticipate there being evidence type differences. What do you think accounts for that?
That is a really good question, and it's something that I didn't expect to see as well. I'm not really sure why this is happening. I don't know if it's the material itself, if certain taxa maybe have a preference for the type of material. I'm waiting to get more sequencing data back to kind of see if this pattern is observed over each month and each location. But I honestly, I don't know. It's very interesting, and I hope that I can figure that out at some point.
Okay, thank you for that. I found that was really interesting. Petra, a question for you. You talked about sort of the differences in trauma as you move away from a blast environment. Did you see any kind of, or do you anticipate, I guess maybe you're earlier in your research, do you anticipate there being sort of a zone outside of that blast area that might, in a way, mimic or be confused with a trauma from a fall?
Yeah, I think there's going to be a level, depending on the size of the blast. That's key to that element as well, or that variation as well. So if you have a really small explosive, like a firecracker or something little, and it's going off right against the body, then you're going to have very isolated trauma in that area. If you have something more like a car bomb or something that's really large, and you have somebody that's standing within like that blast wave radius, that's going to be still in that high impact area, then you could have something that mimics a fall because it'll impact the full body equally. And that's kind of what I'm attempting to do with my experimental data, is to mimic the blast wave impacting the body in a way that would be similar to a fall, just to see if I can start identifying differences that you could see with blast trauma fractures versus fall fractures, because the forces should be similar, but also there's that one's being caused by air impact versus like an impact with the surface. So there should be some differences that are present there as well. So the hope is that there will be some velocity differences that will be illustrated through that research, and hopefully get a little closer.
Yeah, no, thank you for that, Petra. Joseph, a question for you. Using the Ketter's extraction, is that fully optimized? I mean, I'm familiar with it from the point of view of looking at other kinds of materials like explosives or other kinds of different materials. But is there any optimization that could be performed specifically for your particular application, since it's, a little more novel or different?
I did some relatively extensive optimization to bring down the volumes to the size that I was trying to. So there were papers published using liver tissue, using like 5 grams of tissue, and I modified that down to 0.2 and 0.1 around the target size of the insects for the target mass. So I did scale all that down to microcentrifuge level size, so that way I could use the insect tissue. So I did optimize that down to for the tissues.
Okay, understood. I'm just not as familiar with that particular extraction technology. Teresa, I have a question. Have you, or would you anticipate doing any sorts of studies looking at the stability of your eDNA on evidence for its storage? I know you mentioned, the stability, but oftentimes evidence is stored for a period of time before it might be evaluated or addressed. Do you have any idea if that storage time, especially if you're looking at a cold case where that eDNA might be, several years or maybe even decades old?
So there has been one study that I can recall that has looked at specifically bacteria and different storage conditions. To my knowledge, I don't think there has been one with respect to fungi, arthropods, and plants, but that is something that our lab is currently pursuing.
Okay, very good. I don't have any specific questions right now, but I'll press on with just a few more of my questions that I just thought of as I was listening to presentations. And that is, Joseph, I think you had talked about, or you had been asked about drug combinations. Did you, in your authentic sample, you said you didn't have any idea about whether or not there was any sort of like polydrug, effects on the larvae there, correct?
Yes, I analyzed that. I analyzed that liver tissue with the a method that I developed for 34-fentanyl and fentanyl analogs. So and I had fentanyl, 4-MPP, and norfentanyl in that tissue, but it didn't have any other analogs in it. However, I don't know if it may have had, say, morphine or amphetamine or anything like that.
Okay, so you don't know if there were potential additional effects from other kinds of drug types, correct?
Yes, outside of that 34-fentanyl.
Okay. And that authentic sample, that was just a single sample, is that what I'm understanding?
Yes, like it was a single liver specimen.
Okay. All right. Teresa, we have just another few more minutes. You mentioned taxa compositions results for plants, bacteria, fungi. What about arthropods?
So that's a good question. With the arthropods, we were having a bit of trouble capturing or really recovering sequencing reads. We can see that there was amplification, but we were having just some difficulty trying to recover those reads. And so actually, I just got some data back from our core facility yesterday, and we tweaked our workflow a little bit for CO1 for arthropods, and we recovered our sequencing reads. So hopefully, within the next couple of weeks, I can analyze that data and have similar figures as to what I presented today.
Okay, great. All right. So with that, I am going to call our session closed. So I don't think I have any more new questions. Let me just check. Nope. All right. So what we're going to do is go ahead and start our second session. But in the interim, if you have any additional questions, feel free to reach out to our presenters. Their emails will appear on the slide following our Q&A session today. Thanks.
Our first presenter of Session 2 is Sana Albers, a doctoral candidate at the University of Washington. She will be presenting matched probabilities for next-generation sequencing data of forensic autosomal STR markers. Without further delay, here's Sana.
Okay, welcome everyone. Thank you so much for being here and listening to my talk. My name is Sana Albers. I am a PhD candidate at the University of Washington, and I will present some of my work today on match probabilities for sequence data of forensic autosomal STR markers. So first, to give a short introduction of forensic DNA interpretation, it has been centered on the analysis of short tandem repeats or STRs. Traditionally, we use a method called capillary electrophoresis to gain access to the allele numbers that are contained in a sample. Now, with sequencing methods, we have added a new addition to the field, or a new dimension to the field, because we can now see the actual sequence underlying an allele number. So here's just an example of a couple of loci. So for D3, for example, we know we have an allele number 15, so that corresponds to the length or the number of repeats. But when we look at the sequence, it actually turns out that there are different sequences underlying that allele number. So sequencing methods have helped us to see more variation. And that obviously when we are going to change or if we want to move to sequencing methods, we need to check all of the things that are in place, right? So all the software, all the statistical calculations, we need to reevaluate those. So what we're going to focus on right now is match probabilities. And before we go into that, I want to take a quick second to talk about the difference between profile and match probabilities, because that hasn't been always clear. So when we quantify the strength of genomic evidence, there are several questions that we can ask ourselves. The first one is, what is the probability a person has a particular DNA profile? A slightly different question is the second one. What's the probability a person has a particular DNA profile if it has been seen once? The first one is the profile probability. It's interesting. It's usually really small, but it's not always of relevance. What's most relevant in a forensic setting is usually the second question, and that's the match probability. This probability is usually slightly bigger than a profile probability, and that's just the genetic essence of DNA, right? If we've seen a profile once, it increases our chances of seeing it again. So I will be talking about match probabilities, and when I'm using the term match, I'm talking about comparing either two profiles between individuals or from like a crime scene sample and a suspect. Okay, so how do we go about calculating these? We need some data. I have a set of about 1,400 individuals. I have both sequence as well as length data, of course. If we sequence it, I also know the allele numbers. And this is just an overview of the genotypes for one specific locus. I selected this locus because it has only a couple of alleles, so that allows us to do the calculations by hand. So here's an overview of all the genotypes based on length that I saw in my data set. So for example, I have 55 individuals that are homozygous for allele 6. Now what I can do is I can calculate the observed number of matching in my data set. So I know out of those about 1,400 individuals, I can just compare every pair of individuals. So I take one person and then I compare to all of the others. So I have about 2 million pairs of profiles that I can compare. Now, out of those profiles, how often did I find a match? Well, I can go over all of these cells in this table. So those 55 people who are homozygous for type 6, they all match to each other, right? So I take one person from that group of 55, they match to all 54 others. And I can do that for all of these cells. So I'm basically going to add up all of these matching proportions or matching numbers over all of these individuals and pairs. And I will see that I get about 140,000 matches. So if I take the ratio, I get an observed matching proportion of about 0.0742. Now, I can do the same exercise, but then when I use sequence data, it's slightly different. Here, it's still the same locus, SO1. I selected it specifically because it's easier to do because I only found one additional sequence variation. So I have allele of length 9 repeats, but I found 2 different sequences of that length in my data set. So I had one individual who had this genotype with 9 and 6, and I had another individual who also had this rare sequence of type 9.39. So what happens is actually when I look at this data, my matching proportion is slightly lower, right? Because I initially counted this one person as matching to all those 101 other individuals. I did the same for this one person. I thought it was matching to 72 other individuals when I look at the length-based allele. But when I look at the sequence, they actually do not match. So I've counted a couple of matches too many. So if I take the ratio in this case, my matching number of matches is 142,034, so it's slightly lower than what we just saw before. And my matching proportion becomes 0.0742. So it's just slightly less than the 742 that we saw before. But it does show that sequence variation will lead to smaller matching proportions. And this is not necessarily new information. We already knew that this was happening just because we can observe more sequence variation. But it will be interesting to see how it changes and how it impacts our results. Okay, so what we can do is if we have observed matching proportions, we can also predict matching proportions. And we can do that based on population genetics theory. I'm not going to show you the math involved, but here you can see some results for single locus results. So I have a couple of loci listed here that are in my data set, and I calculated the observed and the expected match probabilities for both length-based, so this is just allele numbers only, and I'm ignoring the sequence, and sequence-based results. And as you can see, the expected number, it matches quite well with what I'm observing, right? So this seems to work quite well. Sometimes I'm overestimating the matching proportion a bit. Other times I'm underestimating it, but overall they seem to match quite all right. Where you can also see, so for THO1, this is the one that we just calculated. So this is the 742 and the 740. and these are the corresponding expected probabilities. Another interesting thing I think from this table, and of course it's just a subset of the markers that I have, but for D12, for example, this is one of the markers that shows a lot of additional sequence variation. And you can see that coming back here. So the observed matching proportions, when I look at the allele numbers, is 0.028. When I look at the sequences, it goes down quite a bit, actually. Now, this is interesting, but what we're really interested in for a forensic setting is multi-locus matching, right? The power of DNA profiling comes from using many loci. And here are just a number of matches, or the number of matching loci that I found in my data set of the about 1,400 individuals. I have a total of 27 markers, and I just look at each combination of people. So for each pair, I'm just going to keep track of how many markers they match at. So most people match at only 1 or 2 markers. A couple of them match up more. In my data set, I found even one pair of individuals that match at as many as 11 or 12 loci. So that's quite a bit. But you can see it decreasing, of course, as we would expect. You can also see for length-based in solid line, and then for sequence-based in the dotted line, you can see that for the sequences, it's slightly lower, as we expect, right? There's more variation, so there's slightly less matching. Another thing that you can see, so this is on a log scale. So if matching over a loci will be independent, I would expect this to be a linear line, but it's not, right? We can see that the more loci we include, it's decreasing more steeply. So that has some effect on our calculations. So how does that look like? Well, here are the results when I just look at length-based results. I'm ignoring sequences in this case. And I calculated multi-locus probabilities for 2 locus combinations, 3 locus combinations, 4 loci, and 5 marker combinations. And I plotted the observed, so this is just by counting what we've done in our data set, and I plotted that against my expected match probability. And my expected match probability for multi-locus marker is obtained by multiplying over single markers, and that's what we call the product rule. This is common to use in forensics. You can see this solid line or the identity line. If my expectations were working pretty well, they would all be around this line, and it works quite well for 2 locus markers. But what you can see is the more loci I'm going to include, you will see that more points are going to lie below the line. So you can see that most of the points are below this line. And that's when the observed matching proportion is actually higher than my expected match proportion. So I'm overestimating the evidence in this case and making it out to be more rare than it actually is. And that's not conservative for a forensic standpoint. So that's what we usually want to avoid. So this is for length-based results. We were interested in seeing what happens with sequence-based results. We still expect to see the same trend happening, but is it getting worse or not? It turns out that it actually is. So here is just one result where I'm comparing length-based matching proportions versus sequence-based matching proportions on the right. And I just selected for locus matching. And what you can see, I think there are 2 things that you can see here. The first thing is that for sequence-based, all my points are more in this bottom left corner, which makes sense, right? Because more variation, so my matching proportions are smaller. And the second thing, and it's not really clear, but I made a calculation. I just calculated how often my prediction is conservative. So how often my expected is bigger than my observed. So how often the points lie below or above this line. And for length-based data, it's about 44%. So more than half of the cases, I'm overstating my evidence. For sequence-based results, it's actually a bit worse. I am only conservative in about 33% of the cases. Okay, so this is something that we kind of already suspected, I guess. And we have a workaround for that. So we have what is called a theta correction, which is quite common in forensic calculations. Basically, what I'm doing here is the expected match probabilities. Instead of just using population genetic theory and writing out an expression based on allele frequencies, I'm going to incorporate a theta value, and that theta value can account for multi-locus dependencies. So what I've shown here is length-based versus sequence-based results again for 5 locus matches in this case, and I'm using different increasing theta values. So let's say the red or orange kind of dots, those are when I'm not using any theta value or I'm just setting it at 0. So that's the same as that we've seen before. And I'm not really conservative in these cases. But when I increase my theta value to 0.5%, 1%, 3%, 5%, you can see all of those points shifting upward, right? And then for these purple dots where I'm using a 5% theta correction, most of them are above the line. So if we look at the numbers actually here, you can see them. So for length-based results, the proportion of being conservative changes from about 29% when I'm not using a theta correction to about 88% when I'm using a 3% theta correction, which is kind of standard for forensic calculations. But if I use a 5% theta correction, it actually increases to about 98%. So that's pretty nice. For sequence-based data, it's slightly less conservative. So if I'm not using a theta correction, I'm only conservative in about 12% of the cases. And if I increase my theta to about 3%, I'm still only conservative in about 65% of the cases. So there's quite often when I'm going to underestimate the matching proportions. If I'm going to increase my theta value to 5%, I'm going to be conservative in about 90% of the cases. So what does this all mean? I think there are a couple of takeaways from this. First, the more loci we include, the more we underestimate true matching proportions with the product rule. That's not new information. We already knew that this was the case. What is new is that this is showing that results actually seem to be less conservative when we're using sequence data when we compare it to length-based data. And what we can do to accommodate multi-locus dependencies is invoke a theta correction, so that can compensate for the things that we see here. And I think that will be useful to know, or when we're going to change the sequence data, that we want to account for this. And maybe we even need to account for it by using not just a 3% value, but increasing our theta correction a bit to using a 5% value to be a bit more conservative. Okay, that's all that I have for today. I want to thank NIJ for giving me the opportunity to present my work and for funding my work, and also NIST for providing us with the data. Thank you so much for your attention.
Our next presenter is Dr. William Feeney, a research scientist at the National Institute of Standards and Technology, presenting on the evolution in the development and understanding of GSR analysis via LC-MS-MS. Take it away, William.
Good afternoon, and thank you for attending my talk. My name is William Feeney, and today I'll be discussing my efforts and my group's efforts at West Virginia University for diving into the understanding and development of a GSR analysis method using liquid chromatography tandem mass spectrometry. Now, a special thanks goes out to the National Institute of Justice for funding this work. And so just as a small disclaimer, the opinions and findings and conclusions are that of my own as well as my co-authors and do not reflect that of the Department of Justice. Just for a little bit of housekeeping, whenever I'm referring to gunshot residue, I'm really referring to the conglomerate of two different types of constituents, the IGSR particulates and the OGSR compounds. So after a combustion event occurs or a discharge event occurs, we really form a plume surrounding the firearm, in which consists of these IGSR particulates and these OGSR compounds. Now, these IGSR particulates generally originate from our primer and our casing of the ammunitions, whereas the OGSR analytes primarily originate from the propellant and the lubricant of the ammunition. Now, within that plume that's generated, it's really a heterogeneous mixture consisting of various chemical forms, such as some sulfates, carbonates, and even oxides. Fortunately, the GSR community as a whole right now has this ASTM E1588-20 guideline that highlights scanning electron microscopy, energy dispersive spectrometry, SEM-EDS, to analyze samples that may or may not have GSR on them. So it really does this in two separate ways. One being the elemental side, which consists of varying levels of confidence, and the second being this morphological aspect, which really looks at a spheroid or a spheroid particle that can range from size from anywhere from 0.5 to 5 microns. Now, up until recently, this was the only guideline set in place, but the OSAC subcommittee, the GSR subcommittee, started to focus on really formalizing two different categories for OGSR compounds. But first, let's look at the ASTM guideline here. Some of the key aspects that are good, in my opinion, is that it does provide a very in-depth guide to GSR analysis using this instrumentation, because this technique as a whole is very minimal to non-destructive, depending on who you ask. Now, it does have some key outlining into which you increase your level of confidence with a sample containing GSR based on different combinations. So let's just look at the elemental composition, for instance. So in traditional ammunition, you would typically expect to see antimony, barium, and lead. And so if all of those key elements are present in a particulate, then that sample would have a particle characteristic of GSR. Now, if we omit one of these elements, then we drop down the level in confidence in saying that the sample contains a particle consistent of or consistent with GSR. And then if you omit two of these elements, you drop down further into commonly associated with GSR. Now, some of the limitations that I see with this guideline is that it does only observe one constituent, so just the IGSR side. And to me, at least, I think the term spheroid is relatively vague, because would this particle over here to the right be less spheroid than the one immediately to the right of this, or vice versa? And then as an additional challenge, we are starting to see manufacturers shift into newer formulations, which are termed as non-toxic or even lead-free ammunition. And with that, they're emitting this heavy metal here, lead. And since they are doing that, we are automatically dropping down in a confidence level from characteristic of to consistent with. So that's always a key feature to really keep in mind. Now, shifting into the organic side, as I mentioned before, there have been a couple of research articles that have really outlined separate criteria for identifying a sample possessing organic compounds. But really, there have been just minimal efforts into just the overall OGSR classification. So I think it was around 2020 which the OSAC GSR committee outlined and really placed different molecules in two separate categories using gas chromatography and liquid chromatography. Those two categories are really outlined here, but the main difference is that in the category 1, these compounds that you would see over here to your right are uncommon in commercial products, whereas these compounds here below, you would typically see them in commercial or environmental sources. So that's really the main difference between these two. And just like the ASTM E1588 guideline, this whole categorical system would also be sensitive to different types of formulations presented by the manufacturers. So that kind of brings us to where we started to get into this realm. And so just as your tidbit of information, here's just a general breakdown of the past 20 years of research articles. So within this work, roughly 61% of the reported literature only look at IGSR elements as a whole, whereas only 30% of those reported literature focus on just the organic side. Now, it's really interesting that we did see a upsurge of a kind of combined analysis, which accounted for 10% of this. And so those studies outlined in this work here really focused on the general composition, collection protocols, transfer, and persistence. So where we wanted to really contribute to the community is we wanted to focus on this 10% of combined analysis. But we first had to ask ourselves which analytes did we want to use. Well, at the time, we wanted just to focus on traditional ammunition using the ASTM elemental composition as our set point. So that would be for the IGSR side, and for the OGSR side, we wanted to choose analytes that were commonly reported, and conveniently, we were able to cover both categories presented by the OSAC committee. So those analytes are listed here. So for the OGSR side, we have acridite, ethyl and methyl essentialite, diphenylmine, and its derivatives or degradation products. And for the IGSR side, again, we have lead, barium, and antimony. And since LC-MS-MS cannot really give us these morphological features here, we wanted to focus on just the elemental compositions of these three elements, or those isotopic distributions of those three elements. So how did we approach this? Well, just like with any type of method development, right, we wanted to first to see if it would work. So we wanted to use and focus on just from a single sample, perform a series of extractions of organic and acid extractions. And we took a small twist in the use of complexing agents. And fortunately for us, they were able to not really interfere with our organic signatures and our organic results. And I'll go into further detail on how that actually works here in a few. But from that proof of concept work, we had to optimize our source conditions via electrospray ionization and really focus on these metal characterizations using high resolution mass spectrometry or Orbitrap. So once we optimized it, we validated it using rigorous testing and using the figures of merits, which really looked at the limits of detection, limit of quantitation, as well as these other key components. From here, we used standard mixtures from AccuStandard or from other types of commercial sources. Now, to really test the performance of our method, we used authentic samples. And so since we wanted to really focus on this dual detection part, we had to develop our criteria. But there's an overarching question always into which that would quantifying in this case be crucial. I mean, to an extent, yes, but we're more so focused on if gunshot residue is present or absent on a stub sample. So now we're going to get into some of the results. So here we saw an improvement from our proof of concept study in which we improved the LOD and LOQ, and we were able to increase our target list based on that review article. And by using a different column and retaining the same solvents, we were able to decrease our analysis time to less than 5 minutes here. Now, where I think the cool part really comes into play is this complexation. And so from that single sample, we did a series of organic solvents and acid digestions. And so from those acid digestions, we had to dry it down to really pre-concentrate our metals. But we were really concerned with whether or not pH would be a problem, as we wanted to preserve our electrospray ionization source. Well, by diluting those dried samples with our complexing agents, we were able to protect our ESI source, and our complexing agents actually can ignore the pH, unlike other chelating agents. So whether or not pH was a problem, it was not. And an additional benefit from these complexing agents here is that it allows us to really push our solubilized IGSR particulates through our column to be ionized via electrospray ionization. So after we solubilize it, our metals really fit snugly into the cavity of our complexing agent here. Again, these complexing agents are able to be ionized via electrospray ionization, in which we can now kind of filter out other types of contaminants that may be there using our first quadrupole. We could fragment to push onto our second quadrupole. We can then fragment this complex and really separate out our metal to illustrate its natural isotopic abundances that you see here. Now, although we get these added benefits, we can also retain our natural isotopic distributions, as you can see here. So this would be for barium and this is for lead. And the just simple addition of these simple complexing agents, we're just able to increase our M/Z values. So we were able to really confirm that. But as with any method, you always want to test via its performance using authentic samples. So again, we want to really tailor this towards a dual detection methodology and tailor it for a confirmatory test for a dual detection methodology. So we had to develop our own criteria in which a sample would be called positive for a gunshot residue if it had present 2 organic components with 1 IGSR element or signature, or vice versa. And since we're analytically driven always, we had to have all the signatures greater than or equal to a signal-to-noise ratio of 3. So here are the results of our controlled settings. So we discharged 5 shots from a firearm, and here are the analytes again listed here from session 1 and session 2. And as you can see here, from the first set, we had a low of 90%, and in the second set, we had a low of 44%. And for the first session, we used 1 ammunition, and the second session, we used multiple ammunitions. So that's the key difference. But if we combine both the OGSR and IGSR constituent data, we're able to see that increase from that low of 90% in the first session to 95%, and that low of 44% to 100% in session 2. Now, this, again, was in a controlled setting, so our WVU ballistics laboratory. So we wanted to really mimic more real-world scenarios. So we performed a series of activities in which we, after a discharge event, we performed vigorous hand rubbing, we applied hand sanitizer, and we ran for roughly 60 seconds immediately after discharging a firearm. Now, another key factor that we did not account for is that with this running, we had to really make sure that the weather was on our side, and in the first session it was not. So here you're seeing the results with rainy weather, and the second session here you're seeing the results of more favorable conditions. But really, all in all, this captures that really with one constituent analysis, you're dropping your evidentiary value. But with both constituents, you're increasing your confidence in that sample containing gunshot residue or not containing gunshot residue. And as I mentioned previously also, that the shift in formulations do factor into the observation of your key components here. So where we took that project further is really kind of a population analysis in which we wanted to move more away from our categorical approach and move more towards a probabilistic approach in which we had a large population of bystanders, those activity samples that I mentioned earlier, and baseline shooters, compared to our background samples, which would be high risks individuals and low risks individuals. From then, we would be able to gather all those signatures and be able to perform a machine learning algorithm and generate a probabilistic output, which can then be translated into likelihood ratios. And really the purpose of this would be to more so assess the weight of that evidence when presented in a courtroom setting. Now, with these likelihood ratios, again, just said that we were able to assess the weight of the presented evidence, given that we have presence of both components and the concentration levels in those combinations. But more so, we wanted to objectively screen a sample for gunshot residue here. And with this all in consideration, right, we wanted to really objectively separate a shooter versus a non-shooter, an activity sample versus a non-shooter, or even a bystander versus a non-shooter. And just as for your added knowledge here, we use neural networks for the generation of these likelihood ratios. And I know I went a little bit over time, but just as a key for you as to leave with, where do I see this going? First things first, we are seeing a shift in more formulations, and so we need to start shifting also our targeted species into different non-toxic ammunition and start really incorporating that OGSR into our detection methods. Second thing is that I feel like we can have quicker analyses or screening techniques at the front end, such as electrochemistry or laser-induced breakdown spectroscopy, and then continuing those same samples onto GC or LC methods. And then with this type of approach, and even with the incorporation of these screening methods, I feel like these could be very simple additions into trace analysis groups specifically. So I know I'm over time, but I thank you all for listening, and I'll be happy to take any questions at the end. Here's my contact information, and thanks again for the NIJ for funding everything that you see here, and thanks specifically for these individuals as well as my research group for helping me along the way.
Our final presenter of Session 2 is Kiana McKenzie, a graduate researcher at the University of Louisville, presenting her talk, Generating Representative 3D Pediatric Healthy Femur Models for Biochemical Analysis. Here is Kiana.
Hi, my name is Kiana McKenzie. I am a doctoral candidate at the University of Louisville, and today I'll be presenting on generating representative 3D pediatric healthy femur models for biomechanical analysis. This project was supported by the National Institute of Justice. The U.S. Department of Health and Human Services estimated that approximately 102,000 children were physically abused in 2020. In cases of physical abuse, bone fractures are the second most common presentation. But when encountering children with fractures, it can be difficult to delineate between those that are accidental or non-accidental in nature. For example, femur fractures, which is our focus today, are both a common fracture type in young children, as well as more likely to be due to non-accidental injury mechanisms in non-ambulatory children. Therefore, in distinguishing between accident and abuse, it is important to be able to assess the compatibility of a fracture and a stated cause of injury. Research in this field has aimed to help make this determination in determining the compatibility between an injury in a given scenario. And you can split this into two main categories. First being clinical case studies, which are mostly retrospective studies analyzing the difference between abuse and accidental cases, and also biomechanical testing. However, currently is little known about the fracture potential of pediatric femurs through biomechanical analysis. An advantage of biomechanical testing over clinical case studies is that it does allow for specific evaluation of different injury mechanisms. Some common methods for biomechanical testing include testing of pediatric bone specimens, which are limited by the scarcity of tissue specimens available, especially if you're looking under 3 years old. Another common method is the use of ATDs, or anthropomorphic test devices. These are more commonly known as crash test dummies, and you can see an example of this on the right here, where these can be used to simulate injury mechanisms. So on the right here, you have an example of a ATD being used to simulate a short distance drop. For example, if a caregiver had accidentally dropped a child, that's what this mechanism is simulating. However, the use of ATDs are limited by the options in ages and sizes, such as one line of ATDs only being available in 6, 12, and 18-month options, as well as the biofidelity, or the ability to mimic the response of a human child in these scenarios. The final method that I want to summarize for you today is finite element analysis, or computer simulations, which allows for subject-specific evaluations for given loading conditions. This utilizes subject-specific bone geometry, which are represented by finite element models, where these finite element models are often derived from CT scans. However, there are ethical limitations of performing clinically unnecessary CT scans on children due to the amount of radiation that they would be exposed to. An alternative method to using these FE models that are derived from CT scans for FEA is to use a statistical shape model, which provides a means of generating representative models for biomechanical evaluation when appropriate imaging is unavailable. So what is a statistical shape model? It is a mathematical model representing shape variations of a population, which can be used for predicting the shape of the femur based on subject characteristics. So how do you develop this? Well, first you need your input sample population of femur geometries, which you can then use to characterize the shape of that population. You can do this using a tool such as principal component analysis, or PCA, which can reduce the dimensionality of a data set. So what does that mean? In other words, we can characterize the shape of the femur by summarizing the nodes of the femur surface across the entire data set with a much more limited set of variables. So, for example, if you have 10,000 nodes or points representing the femur surface across your entire data set, you can now use PCA to represent it with maybe 100 variables instead of those 10,000 points. In forming statistical shape model and the outcome, which is the outcomes of the PCA, you have the principal components, and these are our new variables that characterize the femur shape. This part is subject independent. And the second aspect are your principal component scores, or the subject-specific scaling factors for each principal component. And we can use these PC scores to relate to the subject characteristics to be able to build our predictive model, where our predictive model would allow for knowledge of only the subject characteristics to be able to generate a representative femur model. So the purpose of this study is to be able to do just that. Our long-term goal is to be able to generate representative femur models for infants and young children aged 0 to 3 years old for biomechanical analysis. To accomplish this, we aim to develop a statistical shape model characterizing pediatric femur shape and use that to develop a predictive model to generate representative 3D pediatric healthy femur models for healthy children based on known child characteristics. Our data set was composed of a total of 96 CT scans of infant descendants, which were obtained from the University of New Mexico's Radiology Pathology Center for Forensic Imaging. You can see the histogram of the available CT scans that we collected from them below, where you can see that our data set is a little bit more biased towards the 0 to 18-month-old range. And in red are the number of female subjects, and black the number of male subjects. To be able to characterize the shape, it's important that we have a similarly meshed model for all femurs in our data set. What this means is that each model must have an equal number of nodes as well as an equivalent location of those nodes. For example, the node at the most superior point on every single femur is labeled node number 1 on every single femur. To accomplish this, the left femur was segmented for each subject, and from this, a 10-month-old was selected to be able to develop our baseline mesh model, which is our template mesh that will be applied to all the other femurs to satisfy that equal number of nodes and equivalent nodal location requirement. You can see that baseline mesh represented here on the right. It is composed of triangular elements with a total of 9,864 nodes. To be able to apply the template mesh to the other geometries, the baseline mesh was morphed to each of the remaining femurs. And to be able to accomplish this, it is first important to be able to identify anatomical landmarks, which will be used to identify similar geometric features between the baseline and target geometry. To accomplish this, we identified 29 anatomical landmarks that had captured the main features of the femurs. And you can see an example of this on the right with the white dots on the femur representing some of our anatomical landmarks that were selected, such as at the proximal metaphysis, you have the greater and lesser trochanter. You have aspects of the curvature of the diaphysis being captured. And you have, for example, at the distal metaphysis, the most medial and lateral aspects of the distal condyles. Now we can morph the baseline mesh to each of the remaining femurs using a software called RBF Morph, where the output of this process are 96 similarly meshed femur models for ages 0 to 35 months. Now we can finally characterize the shape using principal component analysis, which was conducted in MATLAB, where upon importing it in MATLAB, each femur was first aligned to the baseline using Procrustes analysis. To form our test set, 17 femur models were reserved, and these were used to evaluate the model's predictive capability. To form this test set, one model was randomly selected from each 2-month age interval to provide an equal distribution across our age range to be able to evaluate the model's predictive capability. The remaining 79 femurs formed our training set, which was used to develop our statistical shape model, where the statistical shape model was developed by performing a principal component analysis on that training set. The outcome of the principal component analysis, if you remember, are the principal component scores, which characterizes the femur shape, and that part is subject independent, and principal components, and then the principal component scores, which are our subject-specific scaling factors. This is what is used to be able to develop our predictive model. To develop the predictive model, a partial least squares regression was used to regress the subject characteristics with the principal component scores, where the subject characteristics that were considered included age, BMI, height, and weight. To assess the model's predictive capability, we use the predicted PC scores for those given subject characteristics included in the final model to be able to generate the predicted femur geometry. This predicted femur geometry is what was used to compare to the actual femur geometry in being able to calculate our errors and assess the model's predictive capability. To evaluate the model's predictive capability, the following were calculated for those in the test set: the goodness of fit, the nodal reconstruction error, where the error here is defined as the distance between the predicted nodal coordinate and the actual nodal coordinate of the femur, and the error in predicting the volume of the femur. For the results, we found that the first two principal components had captured 99% of the variance of the training set, where both were found to be significant in the PLSR. Our predictors resulting in the best model were age, height, and weight. And in assessing the ability to predict the femur shape of the test set, we found an overall R-squared of 0.97 with an overall mean nodal reconstruction error of 1.67 millimeters. Our mean volume prediction error for each individual femur was 10% plus or minus 8.3%. To give you an example of the prediction for femur shapes in the test set, on the right are two examples of predictions where the top is the best prediction of the test set and on the bottom is our worst prediction. In other words, our cases with the lowest and highest mean nodal reconstruction errors. So the red points indicate the predicted femur shapes and the black indicate the actual femur shapes for these two test set cases. Both of these predictions show the ability to capture the general contours of the femur model, where our greatest errors in predicting are in the more subject-specific regions, such as the left trochanter here, which can be a little hard to see, but you see the black points projecting above the red, and then as well as the exact orientation of the proximal metastasis growth plate. Overall, the greatest errors in prediction of the test set geometries can largely be attributed to errors in predicting the femur length, and this can be seen in our worst case prediction on the bottom here, where the general contours are captured fairly well. However, it's that discrepancy in length is really where you're seeing your greatest errors and differences. However, the predicted femur lengths did fall within normal morphological variation of the training set when compared to femurs of similar age. What this means is while we're not able to provide the most subject-specific representation of a femur for the given subject characteristics, we are able to provide a representative femur model that can be used. In conclusion, we developed a statistical shape model allowing for characterization of 3D surface femur geometry of young children, which we used to develop a predictive model with the predictors being age, height, and weight. When comparing to predictive models in the literature, our goodness of fit and nodal reconstruction errors were either comparable or better. Additionally, the predicted volumes and femur lengths for the test set did fall within normal morphological variation of those of the training set when compared to similarly aged femurs. In the absence of CT imaging, our regression model allows for the prediction of representative 3D femur geometry in infants and young children based on subject characteristics to be able to use in biomechanical analysis. So given a subject with the age, height, and weight, we can now use this in a finite element analysis from that generated representative femur model. And we can use this to assess the likelihood of fracture for different potential injury mechanisms, where these loading conditions can be derived using methods such as gait analysis or ATV experiments. Like I had shown you before, this is simulating a child potentially being dropped from a short height. So we can use the femur loading that's obtained from this ATV simulation and be able to use it in the finite element analysis and apply it to that representative femur model. Additionally, when 2D imaging is available, such as radiographs, an improved representation of the subject's femur can be developed using the predicted shape model. This allows for the expansion of healthy geometry to be modeled beyond those that would exist within the training set. Thank you for attending my presentation. If you have any questions, please let me know.
All right, welcome back everybody to the end of session 2. We're going to start our Q&A session now. And I just want to say thank you to Sana, William, and Kiana for this excellent presentation. Really interesting work. All right. For Sana, here we have a question for you. It says, for the loci with fewer common alleles, do they tend to have more sequence variants that aren't detected through CT, through CE-STR analysis, or is there generally less genetic diversity at these loci? So I guess that's a question about those loci like you had mentioned, the one.
Yeah, so that's a good question. It really depends on the type of marker, and I think it helps sometimes to classify them. So if we talk about simple STRs like THO1, the one that I showed, it has a relative simple structure in that you only have one repeat pattern like AATG, and that's repeated a couple of times, so 6, 7, 8, 9. And I think with those markers, you don't have a lot of possibilities, I would say, for adding sequence variation. I mean, perhaps you can add like a base substitution, but I'm not sure that's very likely. So the variation that we see is more taking place in markers that are what we call like compound or complex markers. So you see different repeat regions. So for example, I have an ATCT that's repeated a couple of times. Then I have an ATGT that's repeated a couple of times. And if we have that sort of like compound or complex structure, that's where we allow for more variation, and that we can see with sequence variation. So there you have the same length, but for example, there's a difference in the number of repeats of those different repeat patterns. So I would say that, yes, if we have loci with fewer common alleles, I think I would interpret that as more complex markers. So you already see more variation, and that means that if there's more complexity, there's likely more possibility for having sequence variation. So we will probably detect that in those markers as well.
Okay, understood. As well as you have the ability now with some of the sequencing chemistry to kind of see, you know, substitutions outside of the actual STR region, just upstream or downstream as well. All right, so we'll move on to William. William, here's a question. What is the time that it takes to extract a sample, as well as the IC method to run on time to run on the LC-MS?
Okay, so for the LC side, off the top of my head, it was that less than 5 minutes. For just a single sample, or yeah, I'm just going to assume a single sample. For the organic extraction and for the inorganic microbulk acid digestion, it should be around maybe 10 to 15 minutes. That's including drying down and the multiple washes. So overall, full extraction to analysis time should take you approximately 20 minutes, in my opinion.
Okay, well, that sounds very plausible within a typical workday. So thank you for that. Kiana, here's a question for you. For the statistical shape model of the femur, is it possible to convert this data into a 3D physical model for additional analysis and testing?
It is. You could 3D print it or use any other methods. The problem with this is going to be able to mimic the exact material properties of bone in that reduction. And that's going to be a little bit more complex, especially with this young of pediatric bone. You have a lot less ossified. It's a lot less hardened than adult bone. So being able to combine those characteristics would be a lot more difficult. However, the 3D printing or whatever could allow you to be able to generate different geometric variations. For example, the degree of the proximal metaphysis, like femoral anteversion, varies a lot between 0 and 1 years old, and there's some individual variation there. So you could look at changing that factor and doing loading and seeing how that affects it would be a possibility.
Okay, thank you, Kiana, for that. Sana, back to you. So what is the trade-off of the theta correction increasing the conservativeness of the finding and accuracy? So like, what's sort of the difference the real world applicable difference between, say, a 1% and a 5% theta correction?
Yeah, I think that can lead to a really interesting discussion because I think it really depends on the perspective. So you're right in that, as I showed, we can increase theta values, which means we're more conservative because, but it also means that I'm less accurate in that they are not as close as what we observe as we could possibly get, I guess. So if I'm talking from like a statistician perspective for myself, I can say that I want to be as accurate as possible. So I can adjust my theta value and try to get my estimates as accurate as possible, so close to what I'm observing. And that is fine because sometimes I'm overestimating, sometimes I'm underestimating, and that sort of evens out. So as a statistician, if it works on average, and that usually works fine. The problem, I guess, or the really interesting part is I think that I don't really care, I guess, that it works on average if I'm in a forensic setting. So in a forensic setting, there's a specific defendant who is on trial. So we want to know for this specific instance where that point lies. And if I can't guarantee you that it's not conservative, we may have an issue, right? Because I may overestimate the evidence. And from a forensic perspective, we just want to be conservative. So in that sense, I think we want to advocate for having those values that get us those conservative results, even though as a statistician, I may lose some accuracy. But yeah, it really depends on where your focus is. And I think it's a really interesting point because we can argue on what is the correct theta value in that case. And I don't think I can't really tell you what people are comfortable with, but I think it does show that from a forensic standpoint, I would argue that being more conservative, I think would trump like being more accurate, if that makes sense.
No, I understand. It is, like you said, it's a very different perspective. Like the average versus shifting our sort of perspective to being conservative. Thank you for that, Sana. William, here's a question for you. With the combined IGSR, OGSR, what, if any, other compounds do you think could potentially produce false positive results for the presence of GSR?
Yeah, so just specifically on the organic side, I can think of only like a couple examples. So those would be like having any phthalates that would be present, which are just common plasticizers found in any type of, that could be found in any commercial product, I believe. And then I think another one would be any type of phenols. So in the earlier works that we did at the review, in my proof of concept study, we saw like any hand sanitizers that had any like sense, we could have a signal for like a phenol type group presence. So that's for the organic side. For the inorganic side, and I think the ASTM guideline, that E1588-20, also outlines this, but zirconium could also be, like for non-toxic ammunition, I think specifically, zirconium could be present, which is from like the airbags deploying in like any vehicle situation. And also in that previous concept study, we saw that my advisor at the time, she had a lot of iron on her hands when we were first doing studies. So that could also be like a presence of false positives. So it's really like the whole point of this whole project was to just combine those and evaluate the likelihood of one sample having consistent of gunshot residue as a whole versus background sample or not, right? So yeah.
Yeah, no, thank you for that. All right, so Kiana, a question now for you. Can this model be updated or adapted to predict the likelihood of fracture in children with the developmental conditions that affect their connective tissue, bone growth, and their density?
It really couldn't be updated necessarily. If it was updated, it would be more in the context of adding in bone density distribution across the bone to be able to model that over the years, because you do have that ossification that's happening. But it would more likely that it'd be adapted. So for example, what I'm using this work for and what my ultimate dissertation is really on is being able to adapt this model to apply to osteogenesis imperfecta children, which is a condition that affects the formation and production of collagen in bone, well, in general, but it obviously affects bones quite importantly. So this can result in effects like the bones becoming more bowed. So what we can do is generate a representative healthy model and then morph that to a radiograph of that OI bone to be able to get a much more representative for that child's femur of that more abnormal shape that you would see in children with those conditions. So it's really being able to use this is more of a healthy baseline that could be used to be morphed and adapted to be able to then use for analysis if you have other tools and options for that specific child or conditions that you could adapt it to.
Okay, thank you. Sana, would you recommend that we transition to a theta of 5% for length-based polymorphisms, as well with the addition of so many more loci since the community implemented the theta of 3%?
I think it's a bit too early to already go there. I think, as I said, there's a trade-off, and if we want to be more conservative, maybe it's good. But I think at this point, I would just recommend, or at least advocate for using a theta value. I think it's not mandatory at this point. So sometimes the calculations involve profile probabilities, and it will be good if we do try to be mindful and at least talk about match probabilities and do account for some substructure. I do think at this point, I think the answer is just we need to be careful, and I think we need to wait probably in the upcoming years, because this is just the data that I've been using, which is, about 1,400 individuals. So I think it will be good in upcoming research. People will probably look at this with much more data, and I think that can give us a clearer idea of what will be a good value to use moving forward with sequence data, as well as with standard STR calculations.
No, understood. Yeah, I mean, we've been using the 3% theta for a number of years, right? Maybe it's time for us to reevaluate that, the use of that particular theta. All right, William, how would you determine the stringency of what is or isn't GSR given the new formulations like lead-free ammunition that don't fit into these traditional check boxes for GSR?
Yeah, so as this whole method that I presented here, it was for traditional ammunition. So if given for non-toxic ammunition was present, let's say, that we would have to just go back to the drawing board essentially. So really such new formulations, the metals that or the inorganic side that we would have to look at would be elements such as titanium, zinc, gallium also would be thrown in there into the mix. But yeah, the overall criteria would kind of have to, in my opinion, remain as the whole. Just you would have to have at least one or more in conjunction with those organic sides. So really just by shifting into those specific elements, I think would really capture the non-toxic ammunition side as well.
Okay, thank you for that. I have another question for you, William. You mentioned a PFP column. Do you have the dimensions and particle sizes?
Yeah, it's a PFP pore shell. I think it's 120. Yeah, I think it's 120, and it's 2.7 microns and 2.1 millimeters by 50 millimeters. So I can type those dimensions into the chat if need be. I know I just threw out a lot of numbers, so.
Yeah, that might actually be really helpful. Thank you, William.
Okay, cool. Yeah, no worries.
Okay, I'm just scanning quickly here for any last questions. I do believe that we've asked all of those that have been asked by the audience. And as we're getting ready to conclude, I wanted to just say a few things about our Graduate Research Fellows. These are all presentations. Their research has been supported by fellowships that have been made available by the National Institutes of Justice. These awards were made in 2019 and 2018 and possibly 2020. Yes, in 2020. Unfortunately, NIJ was not able to offer graduate research fellowships for the past 2 years, so in 2021 and 2022. I want to relay that our leadership is very strongly supporting us bringing back graduate research fellowships in the coming years. So we hope to be able, sincerely hope to be able to offer graduate research fellowships starting in 2023. We really want to invest in our forensic research community, and we do this best by supporting the research. On that, we've reached 3 o'clock, and I just want to say again, thank you for everybody for joining us today for our inaugural Graduate Research Fellowship Seminar during Forensic Science Week. We hope to do this again next year. All right, thanks everybody.
Before you leave today's webinar, please participate in our short 5-minute survey. Simply click the checkbox icon directly above the chat to access the audience feedback survey for today's event. Your feedback is critical information that helps us improve our webinars and continue providing the best experience possible. We would greatly appreciate it. Lastly, across the top of the live stream window are links to our social media profiles, including Twitter, LinkedIn, and Facebook. There is also a link to the FTCOE website, the Just Science podcast, and the FTCOE newsletter registration. Like, follow, or subscribe to stay up to date with everything the FTCOE is doing in support of the forensic community. Thank you again for attending, and we hope everyone has a great day.